Let's talk about some hard-to-kill microbes. So here's a list of hard-to-kill microbes. These microbes need special attention to kill, as many microbial control methods do not work for them. Infections with these microbes may require special control techniques, especially if the infection is on or in living tissue, because the health of the host has to be considered, right? So in this list, we have gram-negative bacteria. We have two genera of bacteria, Pseudomonas species and Burkholderia species. Pseudomonas, the genus Pseudomonas and the genus Burkholderia, they are both gram-negative bacteria, but they've got some extra bells and whistles, which makes them even harder to kill than normal gram-negative bacteria. Mycobacteria, like tuberculosis and leprosy, like the pathogens that cause tuberculosis and leprosy. Mycobacteria are not gram-positive or gram-negative. They're something completely different, and they're very hard to kill. Endospores you know about, but also cysts. Cysts are the protist version of endospore. And then we have non-enveloped viruses, which we'll talk about. So there's two different kinds of virus, enveloped and non-enveloped. Non-enveloped is much harder to deactivate. And prions. So we'll talk a little bit about prions. You'll get more of that after the exam, but they are especially hard to. They're not alive, but they're very difficult to deactivate. So I'm going to put them on this list, and we'll talk about them a little bit. Let's start with gram-negative bacteria. This is a review, right? So here's gram-positive bacteria. Here's gram-negative bacteria. Gram-positive bacteria, this is its cell membrane that all cells have, made out of the phospholipid bilayer. Then you have the thick cell wall made out of peptidoglycan. This is not a chemical barrier, it's just structural support. It's to protect against osmotic stress. Gram-negative bacteria, so this thick cell wall in gram-positive traps gram stain, that purple gram stain. Gram-negative bacteria, conversely, has a very thin cell wall to protect it against osmotic stress, but it has a second phospholipid bilayer. This is a second chemical barrier. You can see it's a selectively permeable barrier, much like the cell membrane. So it's got these pores that select what can get in and out. So if you wanted to treat somebody with that had an infection, you know, we talked about beta-lactam antibiotics. So let's say someone has an infection and you want to give them something like penicillin. So that's just fine for gram-positive bacteria. But the problem with gram-negative bacteria is that so beta-lactam antibiotics attack the cell wall, right? So for that to get to the cell wall of gram-negative bacteria, the drug has to make it through this outer membrane. And it's difficult to get drugs through this outer membrane. Ampicillin is a beta-lactam antibiotic, like penicillin discussed before, but with an extra amino acid that helps it penetrate this outer membrane so it can access the cell wall and take effect by inhibiting transpeptidase, which you find out here in the cell wall, or in here in the cell wall. So that little extra amino acid helps get it through this selectively permeable barrier so that it can take effect. Penicillin can't work for gram-negative bacteria because penicillin won't be taken into this outer membrane by the gram-negative bacteria. So it's protected. So you have to treat gram-negative bacteria with different drugs that can penetrate that outer membrane. All right, Pseudomonas. Pseudomonas is an extremely diverse genera of bacteria, and only a very, very small percentage of them are pathogens. This genus includes 191 species, most of which have nothing to do with pathogenicity. However, they are resistant to many antibiotics and disinfectants, so the couple that do cause disease are very difficult to treat. These are gram-negative bacteria, but they have extra bells and whistles. So I want to talk about the genus. I found some interesting things about this genus that I want to share with you. So some bacteria in this genus, these ones, Pseudomonas syringae, they actually are partially responsible for precipitation patterns across the globe because they cause rain to condense in the atmosphere. Ski resorts actually throw snow inducers into the atmosphere to force snow onto their ski slopes. Snow inducers are enzymes that cause water in the atmosphere to condense, and under freezing conditions it falls as snow. The enzyme that they use in snow inducers comes from Pseudomonas syringae, which is found all over the world on every natural surface. The reason this species of Pseudomonas is so ubiquitous is because it completes part of its life cycle in the atmosphere. So it goes up into the atmosphere, completes some of its life cycle, and then it causes precipitation so that it can fall into a new location. This is a totally harmless bacteria, just like most Pseudomonas species are. And I just mention this because a lot of, if you work in a hospital, you hear the word Pseudomonas and you think immediately, I need antibiotics, I need antibiotics. Well, Pseudomonas does live in the soil, but most of them don't cause disease. So Pseudomonas as a genus, most of them are just healthy soil bacteria that will do nothing harmful to you, right? However, Pseudomonas aeruginosa is one well-studied species within Pseudomonas because it is an opportunistic pathogen, meaning it does not always behave pathogenically, but if the host's immune system is compromised, then it might take advantage. And once a person has an infection by Pseudomonas aeruginosa, these infections are really hard to treat with typical antibiotics used for gram-negative bacteria because the porin structure is unique and does not allow passage of chemicals that can penetrate the outer membranes of other gram-negative bacteria. Additionally, they have other enzymes that span both the cell membrane and the outer membrane, and those enzymes allow them to pump out chemicals that one might use to kill them. So this is the main thing. They have these chemical pumps that span both membranes to pump out chemicals that they don't like. So even if you get an antibiotic in, it's just going to pump it out again. And that's unique to this genus. So this is thought to be why they are so hard to kill with many disinfectants. So it's not just that they're resistant to antibiotics, they're even resistant to disinfectants because they can pump the disinfectant out of their body to prevent poisoning. Burkholderia is a closely related genus to Pseudomonas, so they're in the same family. Again, it's a very commonly encountered bacterial genus with few pathogens among them. There is only one important human pathogen, and it is typically, well, maybe two, Burkholderia pseudomallei and Burkholderia mallei. So these are the only two pathogens in this genus, and they are only accidental human pathogens. They don't require a human host, and their path within a human is sort of a dead end for them. But they accidentally have qualities that cause them to be dangerous to humans if they do happen to get into the human body. They usually only infect immunocompromised patients, so people whose immune systems are already not functioning properly. But once someone has an infection with Burkholderia pseudomallei or Burkholderia mallei, the infection is very difficult to treat and very deadly for similar reasons described in Pseudomonas. For this reason, there has been military research into using them in biological warfare. Now, I put up here that this is an obligate aerobe, so they need to be in an oxygen-filled environment, and they also are rhizobacteria, meaning they live on plant roots, on the surface of plant roots. And many species are really good to have because they degrade pesticides. So even though there's this one accidental pathogen amongst them, most of them are really great because they can be used for bioremediation. So other species in this genus can be used to degrade pesticides that have built up in the soil. Mycobacteria. Myco means fungus. And mycobacteria are given this name because of their mold-like growth. So most times when you're growing bacteria in a petri dish, it just makes a slimy layer. This has like these bumps and edges that looks more rubbery, kind of like yeast. This looks a lot like yeast looks when you grow it in a petri dish, and yeast is a fungus. So myco means fungus, so mycobacteria is a bacteria with a fungus look to it. This group includes some important pathogens like Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. Okay. Tuberculosis is so hard to kill that many disinfectants and antiseptics will specifically say on the label if they can kill tuberculosis or not. The durability of mycobacteria comes from the waxy layer on the cell wall called mycolic acids. Note that this is not an outer membrane. So here's the peptidoglycan that you're used to. Okay, this is what all bacteria have for their cell wall. This piece is embedded in that peptidoglycan cell wall. It's a part of it. This is a peptidoglycan cell wall with extra bells and whistles. It is not an outer membrane. It is not a lipid bilayer. Okay? It is just an extension of the cell wall with some extra features. Okay? So these are, the mycolic acids are the biggest part of this. The mycolic acids, this is lipid. It's a wax. Wax is a solid lipid. So you'll notice that like if you wax your skis or you wax your car, it's hydrophobic, right? It causes the water to bead up and fall off. Well, this is a wax. So it's a kind of lipid, but it's not a lipid bilayer, okay? It's just a solid lipid that's embedded into this peptidoglycan saccharide cell wall. Okay, or sugar, saccharide means sugar, right? So remember, peptidoglycan is a disaccharide. It's repeating subunits of two different sugars. Okay. All right, so it's not an outer membrane. It's actually wax built onto the cell wall. Additionally, there are many components of the mycobacterial cell wall which makes it incredibly thick and complex. Mycobacteria are not gram-positive or gram-negative because they don't pick up either of the two gram stains. They don't pick up the pink stain or the purple stain. So these are identified through other types of tests. It is very difficult to find antibiotics that can penetrate this protective layer, this waxy layer on the cell wall, and it is difficult for even harsher chemicals to penetrate. This special type of cell wall, this mycolic acid cell wall, makes tuberculosis so difficult to treat that it is sometimes referred to as the most lethal bacterial infection, even though it only divides once every 1 to 2 days, which is extremely slow growth rate for bacteria. Bacteria are usually replicating or making a new generation of bacteria on the order of hours. This only replicates once every 1 to 2 days. So it's a very, very slow growing infection, but it's still considered extraordinarily lethal because it is so difficult to treat, because of this protective layer which prevents chemicals from penetrating the bacteria. It's a very good shield. Endospores. So remember that endospores are particularly durable to adverse conditions. They allow the bacteria to hibernate or senesce until conditions improve. How durable an endospore is depends upon the bacterial species, but some can last millennia. Many are very resistant to boiling, to high pressures, to disinfectants, and other kinds of microbial control methods. So you really have to know what kind of endospore you're working with to figure out what control method you have to use to destroy it. And just a quick reminder, so the way endospores are formed: first, the cell replicates its DNA, so it's got 2 genomes now ready for division, but instead of dividing down the middle like in binary fission, it's going to divide into 2 equal parts. You've got the forespore and the mother spore over here. So the forespore is going to be let go and then endocytosed by the mother spore. The mother spore's DNA gets degraded, so now you've got the membrane of the mother spore and then the membrane of the forespore inside of it. So you've got 2 phospholipid bilayers here. Then a spore coat is going to form around the endospore, and also a peptidoglycan cell wall is going to be formed between the two. So you're going to have peptidoglycan as well between the 2 membranes. And then so you've got a phospholipid bilayer, a peptidoglycan layer, another phospholipid bilayer, and then a protein coat that forms around that before it's exocytosed. And then you've got the endospore. And then on the inside's going to be desiccated or dried out. All the metabolism is going to come to a stop. It's kind of like cryogenically freezing it. All right, as an example, I'll give you botulism. Botulism is caused by, we've talked about Clostridium difficile enough. Clostridium botulinum, obviously closely related, it's in the same genus as Clostridium difficile. It also forms endospores. So botulism is an example of a pathogen that is very difficult to kill in food because of the durability of the endospores. Clostridium botulinum is a common soil bacteria, but it is an obligate anaerobe. It does not like oxygen. In the absence of oxygen, it creates a toxin that interferes with neuromuscular function. So that means when it doesn't have oxygen, when it's happy, when it doesn't need to be an endospore, it's got no oxygen around to poison it, so it's just doing its thing. And while it's metabolizing, it creates this toxin that happens to interfere with neuromuscular function. It's not doing that to harm humans. It just makes this neuromuscular function is a byproduct of its metabolic activities. Okay, so it's just digesting. That's all. It just happens to accidentally make a toxin that is bad for you. And so what this toxin does if you ingest it is it causes weakness or difficulty breathing or even death from paralysis and lack of oxygen. Now, it can take up residence in the intestinal tract of very young infants, but that very, very rarely happens in adults because by the time we are 6 months old, we have a healthy culture of other microbes in our gut that prevent Clostridium botulinum from taking up residence. Also, the acidity changes as we age to a pH that prevents it from growing. So, the only time it would actually infect an adult is if the adult had some anatomical or functional abnormalities in the gut, or if they're taking antibiotics which kill off the good microbes that prevent botulism infection, much the same as C. diff, right? So it's not very happy. It doesn't grow very well in the conditions of the human body. But so when you get botulism poisoning, it's not that you have an infection, it's that you've ingested the toxin that it made in your food. So if the soil bacteria accidentally gets dug up, it's floating around in the air, in the dust in the air, it's formed an endospore because now it's in oxygen-filled conditions that aren't good for it. Then it settles into a can before the can, like the can of tomato sauce before the can gets sealed. Now the can is, the can's heated up to commercially sterilize it. The endospore is in there so it doesn't get killed. And then it can, once the can cools down, it can germinate. There's no oxygen, it's nice temperatures, it's great for Clostridium botulinum. It's in there eating the tomato sauce, producing this waste product that happens to be toxic to us. We open the can, we heat it up to 100, we open the can, we eat the tomato sauce. If we eat that toxin, then we will be poisoned by it with paralysis. Okay. Now, the toxin is destroyed at boiling point, so all you have to do to prevent that from happening is just, when you open a can, just boil it. This is very, very rare anyway, but if you're worried about it, just boil it, it'll be fine. Because the toxin is destroyed and the endospores can't really germinate in your gut if you're just a healthy adult. Right? All right, cysts. Protists can form cysts, which are similar to endospores in function. Giardia is an example of a protist that forms a cyst. Garlic is actually a little known effective treatment for Giardia because it immobilizes the flagella, disabling the flagella from the Giardia from acquiring nutrients and energy sources that they need and making it easier to excrete them in feces. But the garlic has to be taken for quite a while because the vegetative protists of Giardia, because Giardia is a protist, the vegetative protists will be removed while you're taking garlic, but the cysts will remain protected from the garlic treatment. They'll germinate and they'll create a new generation after the garlic treatment is done. So you have to keep it up for a while. And you know, the effect of garlic on Giardia has been shown in vivo and in vitro. So both in a petri dish and in live subjects, even though it's rarely recommended, which is I find really interesting. I have had Giardia and I've used garlic treatment and it works phenomenally. So I would say if you like to camp and you've gotten this problem before, I would say give it a shot. All right, non-enveloped viruses. Remember that some viruses have a lipid envelope and some do not. So we will talk a lot more about viruses coming up here soon. So let me just talk real quick about viral anatomy. So here's a bacteriophage. This is a virus that infects bacteria. This is what they typically always look like. And I'm just going to point out some features. Here's their genome. So this is some DNA in here. And then this is a protein capsule for it. It just protects it. And then these are also protein subunits that have some mechanical functions. It's like a very simple little robot. If these protein fibers meet up with a bacterial cell, they latch, and this little sharp thing bores into the cell and spits the DNA in. So it's not alive, it just has some automated features. I mean, we know that we have lots of examples in our life of, in our modern human life of more complicated machines than that are non-living. So that's easy to understand, right? It just, if the fibers meet up with a bacterial cell, they bind, and this spits the DNA into the cell, okay? And then this is an example of a non-enveloped virus because it just has the DNA and the protein capsid. We call this whole protein case with automated features and all, we call it a protein capsid. And its function is to protect the DNA, protect the genetic material, and also to help infect the cell. Now, enveloped viruses have one additional feature. So here's an RNA virus. Some viruses use RNA as their genetic material. This is the flu, influenza. So influenza is an RNA virus, so it uses RNA instead of DNA for genetic material. Then here in white is the protein capsid. It's just a simple little hexagon here. And then around that you have a lipid envelope, right? This is, and it's a phospholipid bilayer. You can't really see that here, but this is a phospholipid bilayer. It's taken from the cell as the virus exits. So the virus doesn't make this on its own. When the virus gets formed inside of the cell, and then as it's exiting the cell, it takes some of the cell's phospholipid bilayer with it. So we call this an enveloped virus, and the enveloped virus has spikes on it that help it infect the next cell. COVID-19 is also, all coronaviruses are enveloped viruses. Enveloped RNA viruses, sort of like influenza. All right, so remember that so some viruses have a lipid envelope, enveloped viruses, and some do not. Those that have enveloped viruses are actually easier to kill because the virus relies on this lipid envelope to protect genetic material, but lipid envelopes are fairly easy to destroy with disinfectants and antiseptics. So lipids are less durable than proteins. So an unenveloped virus has everything it needs in its protein case, and those proteins are hard to destroy. For enveloped viruses, much of what they need is part of this lipid envelope, and if you destroy it, they're inactive. And lipid envelopes are just much easier to destroy than protein capsids. Okay, so this part is confusing to students sometimes because yes, if you destroy the lipid envelope, there's still the protein capsid inside, but the protein capsid can't infect a cell by itself. All of the elements needed for infection are in the lipid envelope for enveloped viruses. So they lose this and they lose the ability to infect. So they become inactivated. So non-enveloped viruses are harder to inactivate. All right, prions. We've talked about this a little bit when we talked about emerging infectious diseases like mad cow disease, right? So these are unusual diseases because most diseases require nucleic acids as replicating material, including viruses. But these are proteins which, quote unquote, replicate by converting other proteins to prions. So it's not that they have a genetic code to make offspring. They take already formed normal proteins and convert those normal proteins to prions. Okay. Prions can only very, very rarely spontaneously arise. Typically, you get the first prion by being infected. And in the case of mad cow disease, as we discussed, you have to ingest infected neural tissue, like brain tissue. The exponential growth of prions means that for a long time after infection, no prions nor symptoms are noticeable. But after some critical population has been reached, the prion numbers start skyrocketing. There is currently no treatment for prion diseases, and though they are proteins, they happen to not be destroyed by temperatures that would typically disable an enzyme. And we don't really know why. The only way to really be rid of them is to incinerate them. For this reason, all those infected cows across Europe, the cows that were infected with mad cow disease, they were incinerated.